Okay, so aseptic techniques are important for microbiologists. You have to clean the bench first with 70% alcohol or other disinfectants. Then you work around the flame. You work near the flame. The flame heats up the air, warms up the air rises up, so there'll be less dust and particles settling upon the bench. So if you open up your agar plate, you'll be more confident that there will be less dust, less particles settling on your plate. So you reduce contamination, chances of contamination. Okay, so that's why you work around the flame. So you also hear that you're supposed to flame the mouth, right? Flame the mouth of the tubes. Why? Is it to kill the bacteria on the tubes? Not necessarily. It is also to to heat up the air around the mouth of the tube so that when the air expands, dust don't go in. Okay? So there's a reason why we do all these small steps. This is to reduce chances of contamination. So these are aseptic techniques that all microbiologists have to be comfortable with. Okay? So of course you have to wear your lab coats. You have to tie your hair if it's long, okay, because it's dangerous. We've actually had students who burn their hair, not all of it, a bit, but you can smell. So okay, so you have to be careful. So when we talked here about gram staining, spread plating, pore plating, dilution streaking, and bacterial identification. So gram staining you've done before in your first year, okay? So just follow the steps. You have to heat up usually we just flame the slide once, then make a smear. We make a smear of the bacteria. Okay, when you make smears of bacteria, do you take a lot or little of the bacteria?
Little.
Yes. So
Right.
why? When you take a little bit, then the chances of the stain being evenly stained, the chances of your bacteria being evenly stained is better. If you take too much of your bacteria, the stain you put might not stain the bacteria evenly. Then you'll see that some are purple, some are reddish, Then you might actually report your results wrongly. You'll say it's gram variable. So it's neither gram positive or neither gram, and then you get gram positive, you get gram negative both together, then you report it as gram variable. So that's wrong already. Okay? So that's why you shouldn't take too much bacteria. Okay? Then we will usually heat fix it before we start with the staining procedure. So here I want you to familiarize yourself with the terms primary stain and counter stain. Okay, so the primary stain is the first stain that you place on it. Okay, the crystal violet stains everything purplish. Then you decolorize it. Okay, decolorization is the ethanol acetone solution. So this is the critical step. If you decolorize too much, all your bacteria will be gram negative. If you decolorize too little, your bacteria, all your results will be gram positive. So you have to uh, be careful with this step. Okay, then after you decolorize, you counter stain it. Counter stain is your safranin. So the term for safranin is a counter stain. So let's just say you're working in the lab for the first time, then you're doing a gram stain. How do you make sure that you're doing it properly? You're in the lab, you're asked to do a gram stain. How do you ensure that you're doing your gram stain properly?
Read,
Ask for the supervisor's help.
read the procedure properly, maybe.
Yeah, but as if you read the procedure carefully, you get your results. How confident are you that is correct? If you ask your supervisor also, So how confident are you that the results are correct? When you're
Around 50%.
so when you're working in a lab, we must always have controls. Okay? As I mentioned earlier, even when when you're preparing plate, you have to have controls. Which bacteria will grow well, which bacteria will not grow well, so that you can test out to see whether your plate is being prepared properly. Same goes for staining. Every staining procedures, you will have positive and negative controls. Okay? So that you know whether you have done your staining properly. So if it's the first time you're doing your staining, you do gram staining on both a gram positive and a gram negative control. Then you should be able to see different results. The gram positive being purple, the gram negative being red. Only when you see this, then you're confident, hey, I'm doing it properly, I'm doing it correctly, I'm getting the results that I know I am supposed to get. Okay, so all labs will have control cultures. If you don't have, then you should ask for it. Okay, you can probably ask your lecturers for control, bacterial controls. Okay, because if not, you will never be sure whether you're getting it properly, you're doing it properly. Okay, so controls are important in all experiments. Controls are important. A positive control, a negative control, and a blank. Okay, so the critical step here for gram staining is the decolorization step. Okay, decolorization. So it's by practice, like how much tilt you need, how long do you need to immerse to wash the smear with the decolorization agent. It's all by practice, but when you have your controls there, your positive and negative controls, then you'll be more confident that it is being done properly. Okay. Okay. So this is a normal light microscope. Okay. So if you look here, it's written here how you should use a microscope. Okay. So microscopes are an essential tool for all bacteriologists. If you don't take care of your microscope, you're gonna have a tough time, because if you don't take care of your microscope will be blurry, it will be blur, you'll scratch the lens and all that, and then you cannot see clearly. Once you cannot see clearly, it will make your life miserable, because you can't get proper results, you can't see clearly. So always take care of your microscope well. Okay, of course, all microscopes will come covered, so you have to cover it when you're done with it. Okay, then you know that there's a microscope stage, right? Stage is here. This is the stage. Lower it maximum. Lower it all the way down. Okay, lower it all the way down. Place the slide inside. Place the slide here on the stage. Okay, so once it's there, use the lowest magnification first. Okay, most microscopes will come with times 4, times 10, times 40, times 100 lens. Okay, so you always start with the lowest magnification, lowest objective. What you do is you bring up the stage. Bring up the stage until it's clear, until the image is clear. Once it's clear, turn the objective to 40. Okay, from 10 times, turn the objective to 40. Then move slightly move slightly, adjust slightly until the image is clear again. Then you're ready to change the lens to 100 times. So when you're changing the lens to 100 times, before you change it, place a drop of immersion oil and then turn the lens to 100 times. When you're moving from 40 times to 100 times, do not move the stage already. Okay, you don't need to move the stage. If it is clear at 40 times, when you move it, when you're changing to 100 times, you wouldn't be stretching. If the microscope is aligned properly, you wouldn't scratch the 100 times. Okay, so you don't have to move your stage around too much. You do not move your stage around too much so that it is easier for you to get a clear image at 100 times. Okay, 100 times is clear only with oil immersion. Okay, so a lot of students find it very difficult to get clear images because they don't know where they are focusing, what image they are focusing on. So for me, I find that it is useful to slowly move the slide left and right when you are focusing so that you know you are focusing on the actual bacteria. Okay, hopefully you'll get to try this once the maybe in sem 2. Okay, we'll find a time where we can have some lab sessions together. Okay, then once you finish using the microscope, you have to clean the oil from the 100 times lens using Kim wipes or lens paper. Okay, this step is crucial because the oil will actually degrade the rubber lining around the lens. So if you don't clean the oil properly, it will actually spoil the lens. Okay, so these are steps that you must remember. Okay, so because you have a 20% test on practical theory, so these are some of the things that I may ask you. Okay, so please take note. Okay, where any questions?
No question.
Okay, so for the pore plating enumeration, we can do it another day. Okay, so for bacteria, for colony morphology, so when you're growing bacteria on plates, they grow as a colony. Okay, bacterial colony. So if you look carefully, you can see flat, raised, convex, pulvinate, and umbonate shapes. So these are how a bacteriologist can describe the colony that they observe, whether the boundary is entire, undulate, crenate, lobate, dentate, rhizoid, and filamentous. So you have different definitions for different boundaries that you see. Okay. Okay. So when you looked at the Glassware and Apparatus YouTube, yes?
Yes.
So
Yes.
Yes.
any questions about this? Pore plating and spread plating. So for enumeration of bacteria, there are two. Here you are exposed to two types, pore plating and spread plating. So for pore plating, your sample is placed in the petri dish before you pour the agar onto the plate. So when you mix your agar, when you swirl your agar plate, you are mixing the agar and the bacteria sample together, and then the colonies will grow separately. Okay. Whereas for spread plating, you're using a spreader onto the plate. So you're placing your sample on the plate and then using a spreader to spread it around. Okay. So when you're doing this, for pore plating, your bacteria actually grows inside the agar. Whereas for spread plating, your bacteria will grow on top of the agar. So it's two different techniques for counting bacteria. Have you done this? No. Either one?
Doctor, in bacteriology too, I think we did spread plating.
Oh, but not everyone did bacteriology too.
Oh yeah.
Okay. Okay, so yeah, okay. We'll have to find a way to do this. Then dilution streaking is where you try to separate the bacteria growing into single colonies. Okay, usually what we do is we take the initial colony, then we spread it around, then we flame the loop, then we streak the second time, then flame the loop again, then streak a third time, then flame a loop, and then do the final streaking here. Then that will give you single colonies. So dilution streaking method is how you get single colonies from a mixture. Okay? Okay. Any questions?
No.
Okay. So, have you seen the video, the introduction to glassware and apparatus?
Yes, Doctor.
Yes.
Okay.
Yes.
So the main point is the volumetric flasks are for preparation of standards. Okay, so that is what I want you to remember. When you're preparing standards, especially chemical standards, you must use volumetric flasks. Okay, because all other glasswares are not as accurate. When you're preparing standards, it has to be accurate. If not, you won't get good results. Okay, so that is the main point that I want to share with you. So when you're using electronic balance, it's very straightforward, but you have to sometimes take note that the balance is actually flat on the table. So sometimes you'll notice that electronic balance have bubbles. Okay, so if it has bubbles, then you can make sure that the bubble is placed in the middle. If it's centered, if the bubble is centered, then you can be assured that it is balanced. pH meter, very straightforward, but you must make sure that you calibrate it with your buffer solutions before you use, and to always keep it in solution, the probe electrode. If not, it will dry up, then it'll spoil. For micropipette, just remember that there are 2 stops. Okay, just take your time to look through the YouTube